In lab 2 with the cell, we talked about moving material into and out of the cell. With this figure, we are showing a cell that is taking in molecules. This is taking in, this looks like a bacteria, so it's a large molecule. And when you take in a large undissolved chunk, it is called phagocytosis. And when you take it in, it's phagocytosis is, phago means feeding in Greek. So we'll take it in, engulf it with these pseudopods, these are extensions of the cell membrane, and then enclose it in a vacuole. The lysosomes will then fuse with the vacuole and break down the material if this was a white blood cell attacking a bacteria. And then the debris will now be spit back out of the cell through exocytosis. We will also do pinocytosis, where we take in small dissolved molecules into the cells. Then the last thing is I want to show you some pictures of tonicity and osmosis. Tonicity is the measure of dissolved solutes in a solution. And with this, we're looking at a red blood cell. And this red blood cell is surrounded by an isotonic solution, which means it has equal salt-water balance inside the cell and outside the cell. So even though there's net movement, there is no change. And the cell will start out as a concave disc and stay a concave disc. And this is the environment your cell wants to live in. However, if we take that red blood cell and we put it into a hypertonic solution, which means there is more salts or solutes outside than inside, the water will exit the cell at a rapid rate and the cell will shrivel. And when you're looking at red blood cells, this process is called crenation. The last is when you take that red blood cell from a isotonic solution and you put it into a hypotonic. Hypo means that there is less solutes outside, more water outside, so the water is going to enter into the cell and cause the cell to swell. And if it is extreme enough, the swell will actually rupture. And when the red blood cells rupture, it is called hemolysis.